Good day to everybody who is coming to our ZOOMinar series. We have two speakers today, Dr. Ruggeri and Dr. Ni. However, Dr. Ruggeri is running late, and we will start first with Dr. Ni. And as usual, our seminar series, the speakers have about 30 minutes to present, which will be followed by about 10 minutes for questions to the speakers. And at the end, whoever you want to stay longer, you can ask both. There will be a discussion with the two speakers together. But I don't want to delay any further the beginning of the seminar. I'll pass it to Dr. Wu, who will be introducing our first speaker today, Dr. Ni.
Hello, everyone. Today we have a speaker, Dr. Ruiqing Ni from Switzerland. She's a group leader from the University of Zurich. Dr. Ni obtained her bachelor degree in pharmacy at Fudan University and received her PhD degree from Karolinska with Professor Nordberg for amyloid-beta imaging. Her current research interests mainly in MRI in vivo imaging. Look forward to her talk, Dr. Ni.
So I will share my screen. May I ask if I should swap the display or it's fine?
Go to the presenters mode and then you can get started.
Okay.
Yeah,
is it correct now?
Yeah, it's perfect.
Okay, great. So thank you. So I would like to thank Rams for the kind invitation and Magda and Jinhui for the introduction. So I will mainly talk about non-invasive imaging in animal model for A-beta and tau. So so my research focuses on developing imaging biomarker in neurodegenerative disease, and I mainly work with in vitro probe characterization and small animal imaging. So I will discuss first probe characterization using febrile brain samples, and next we'll present in vivo multi-scale imaging of A-beta and tau with optical and PET imaging method. So we mainly use transgenic animal models such as APP-PS1 Arctic A-beta mice to represent amyloidosis, and tau mouse model with P01L, P01S that with full repeat tau to model the tau-C. So there has been a rapid development in amyloid imaging tracer. 3 of these tracers has been approved by FDA and EMA, and amyloid imaging has demonstrated their clinical utility and validity. So we can see that on the screen, the first row is the PET imaging in AD patients. Second row is the PET imaging in control cases. We can clearly see there are much redness in the cortical region. In the, I should start the point. So we can see there are much more redness in the AD cases compared to the control cases. And many of these imaging probes actually come from the dyes that we used for staining amyloid, such as COP11T and Stilben structure. So in addition, there has been a very fast development in imaging of tau neurofibrillary tangle in patients with Alzheimer's disease and in patients with primary tauopathy. This facilitated the early and differential disease diagnosis, as well as in vivo back staging. However, currently there are still no tracer for full repeat tau. One of the tracer for taucipir has been approved by the FDA. So as you can see on the screen, there are first generation of tau tracer and the second generation of tau tracer. one limitation of the first generation tau tracer is that they have rather high off-target binding, which is improved in the second generation. So we will mainly use the PBB3 compound in my study. And so for the method-wise, so for the in vitro characterization, there has been different in vitro methods used for probe characterization. Depends on the labeling. If it's a radio-labeled compound, we can use radio-labeled ligand displacement assay to characterize the binding sites, and also use autoradiography to characterize the distribution of tau tracer distribution, for example, in the human brain slides. And radiometric fluorescence assay can also be used to assess the binding property of the probe, and staining for ex vivo distribution assessment. For more accurate evaluation, solid state NMR and cryo-EM has been used to characterize the binding pocket of certain imaging probe on the precise position in the fibro pocket. In addition, in silico method has been used to assess the binding site of tau and amyloid-beta imaging tracer on the fibril. Actually, more than 4 binding sites has been characterized, and different tracers seem to be bind preferably to certain binding sites. So in our study, we have been very lucky to use the LCO compounds provided by Professor Nielsen, and we used a compound AOI, for example. AOI is an oxazine-based compound derivative, and we found that in the staining in the postmortem, in the arctic A-beta mouse cortex, we found the AOI overlay very well with the HFTAA. However, as you can see in the panel C and D, there is some mismatch in the QFTA with AOI 97. So AOI 97 and the HFTAA both overlay well in the parenchymal plaque as well as in the CAA, cerebral amyloid angiopathy. So actually, there has been previous study. They suggest that QFTA may be bind to more mature A-beta fibril, and HFTA might bind to more immature fibril. So we think maybe we also can detect this kind of A-beta polymorphism using other A-beta probes. So other binding assay we did before is to detect the different binding sites in the sporadic AD and the autosomal dominant AD cases. So in the left panel, we see that the compound AZD 2184 is amyloid imaging probe. It can detect 2 binding sites in the sporadic AD frontal cortex. However, the competition assay in the right panel shows that the binding value is slightly different compared with the sporadic AD. So there is potentially a third binding site. However, maybe you can also observe that the binding curve is really noisy. We have a huge error bar. So this is the part we are working on now. So one interesting thing regarding the sporadic AD and the autosomal dominant AD is that they are different conformation of A-beta plaques as seen in the panel below by silver staining. So similar observation was detected by a radioligand binding displacement assay on different tau-C patients, such as PSP compared with the AD brain, and we can see that there is a difference in the binding value between these cases and between the human brain and with the tau mouse model. So we want to develop how to experimentally measure more than 2 binding sites on A-beta tau alpha-synuclein fibril with high accuracy. So cryo-EM is of course really accurate. However, it's experimentally difficult. And so for the ongoing project, in collaboration with Professor Roland Rick and Juan Gers, we use the recombinant fibro to study probe binding sites using a high throughput method using the SPR surface plasmon resonance method. So we used A-beta, catein tau, and alpha-synuclein produced by Juan, and to study first different imaging probe, the binding on the different fibrils, and we can see that they, for example, the HFTA, they show multiple peak indicating potentially multiple binding sites. And next, we use the SPR method by immobilized the fibril on the chips, and we can detect with quite higher accuracy the K on K off, and calculate the KD value, and with a rather okay chi-square. So next I will move to the non-invasive imaging in disease animal model. So we will discuss first the microscopic imaging, such as by using PET imaging, and mesoscopic imaging by using photoacoustic imaging, and maybe MI, and at a microscopic level by using fluorescence imaging. So our main question is, how do we detect A-beta and tau with high resolution non-invasively in animal model? So our purpose is to allow longitudinal imaging to follow up the distribution change and to monitor the change after treatment. And different imaging modality has been developed recently, or in recent decades, including MI PET, which provides a whole brain field of view, but relatively low resolution and imaging speed. Confocal and two-photon imaging offer very high resolution, but of rather small field of view. So there has been recent development in the mesoscopic level. For example, the MSOT multispectral optoacoustic tomography has the unique capacity to image at a mesoscale resolution and the field of view with very high imaging speed in comparison to the PET method. So for the PET imaging, we have been previously using amyloid imaging compounds such as ADD2184 to detect the accumulation cross-sectionally in the APP Swedish mutation, and we can see that there are increased signal such as more redness in the 18 to 24 months old transgenic mice compared to 6 months old transgenic mice. And the level is higher compared also to wild-type mice shown in the lower panel. So in the same time, we also try to see if there's some early inflammation going on in this mouse model. And indeed that we find by using monoamine oxidase B inhibitor, depreniol, which indicate astrocytosis, we detect a high accumulation already at 6 months in the APP Swedish mouse compared to older mouse and compared to older wild-type mouse. So yeah, so this kind of indicating an important early role of astrocytosis in this mouse model. So the great value of PET in small animal imaging is that it provides a longitudinal visualization of A-beta accumulation. So in this paper by Brando et al., so they show in the different transgenic mouse model by using florbetaben, they can map the increase along with increasing age, and a different rate of increase of plaque is demonstrated by PET imaging method. Yeah, so we also work on tau imaging by using PBV3 in the RTG4510 and PS19 mice. In addition, there are other probes being used, such as the THK compound and also florbetapir. So as well as in rat model recently. So for the tau imaging, we think, we think there is actually still a gap in preclinical imaging of A-beta amyloid in mouse brain. Thus far, as we demonstrated before, PET imaging has the issue of a resolution, that the resolution is around 1 millimeter, which is rather limited compared to the small animal head. And fluorescence imaging, such as 2D near-infrared imaging, has the issue of scattering and cannot reach deep region. 2-photon imaging can monitor the single plaque growth over the time. However, it is of very small field of view. So we want to focus on the mesoscale part. So far has been several papers published using such method in combination with fluorescence imaging, as well as combination with like triple modality optoacoustic PET and fluorescence imaging. So this has been applied for detecting A-beta, tau, as well as microglia and macrophage. So optoacoustic tomography is actually based on a quite simple principle. So the laser excites the absorber in the tissue, or it can be induced by external kind of imaging probe, for example. And this will cause local temperature change, and the local pressure will rise and propagate, and this will be changing in the ultrasound wave can be detected and used for image reconstruction. So the device I used is developed in Professor Daniel Rozansky's group. So this method can provide a 3D multispectral imaging with a resolution of around 100 microns. Spatial resolution, as you see in the middle of the slide, we can clearly see the kind of PR vessel, and this is with kind of intact skull. So the mouse is totally kind of fine, and it can be used for longitudinal imaging. So the setup looks like this. So we place the mouse under the transducer, and so the mouse is kind of injected intravenously with the imaging probe, in this case AOI-987, and afterwards we detect for about 2 hours and detect like a wash in and a washout, and afterwards we calculate the retention of the probe and reconstruct the image, and then map it on the mouse brain atlas, and then do a mixing into the probe channel and the endogenous hemoglobin channel, and do the calculation of volume-based analysis. So this slide shows. I think I cannot play the video. Okay, sorry, I have some problem on the video playing. So basically, on the left side, we see the mixed channel. So the blue and green indicate the probe or the amyloid distribution, and the orange-ish color indicates the vessel. And on the right side is the kind of co-registered image of amyloid distribution detected by MSOT with the mouse brain MI structure, and we can use it to see the kind of 3D distribution of amyloid in the mouse brain. Yeah, so basically, we apply this in 2 mouse model, APPBS1 and Arctic A-beta mouse, and we can detect more accumulation in the cortex, hippocampus, and the thalamus compare the transgenic mice with the wild-type mice. And we applied the same method using another compound, Quinase2, which is a curcumin-based compound. And basically, we detect the same kind of pattern. And so the advantage of this method is that it is non-invasive and can be used for longitudinal imaging. So I have to point out we have a very short longitudinal study. Ideally, we will extend it, but nevertheless, we can see there are changes, increase in the cortex region compare 15 months to 14 months aged transgenic mice. And so other methods have been developed. So the FMT system is a fluorescence molecular tomography system developed by Professor Marcus Ruiqing. So this method actually can also be used to detect the 3D distribution of molecules by similarly injecting the amyloid imaging compound into the tailbone of the mouse, and we can reconstruct the distribution of amyloid plaque in the mouse brain. However, this method is of a lower resolution compared to what we showed before. And we applied a similar concept to tau imaging. So I will go through quickly. So basically, we also see a gap in whole brain high resolution imaging of tau in mouse model based on our previous study in 3D tau PET imaging. We found we can further improve on this. Although there has been existing methods using 3D MRI, for example, we see like such method does not provide a very high specificity or sensitivity. And the limitation of 2D near-infrared imaging and 2-photon we have discussed before. So they are limited by either intense light scattering or small field of view. So in this project, we used a compound PBB5, which is derivative of clinically used PBB3. And we can see that upon binding to tau, it shows a higher kind of excitation. And the panel to the right shows that it overlays well with the PBB5 stained tau accumulation in both CBD PSP, which is tau-C patient brain, as well as in PC1L mouse brain, indicating the probe is quite specific and sensitive. So, so this study was done by PhD student Patrick and Arthur. So we can see that compared to the wild-type mice, the PC1L tau mice has a higher accumulation. For example, at 60 minutes, we consider as a retention time in the hippocampus and in the cortex compared to the wild-type mice. And we also follow simultaneously by fluorescence imaging, which kind of indicating the same pattern. So for the microscopic imaging, there has been previously a lot of study performed by using 2-photon imaging that monitor the accumulation and increase in size of amyloid plaque. And we used a method called a large field of view imaging. So basically the concept is to capture the whole hemisphere of the mouse brain without opening the skull. Yes. So basically, we use the compound LCO HS1689, and we can see that so it provides increased fluorescence upon binding to A-beta 42. And so by using this method, we can differentiate, like we can reduce or deduct the background by using a certain filter method. And afterwards, we can see the so we can see the green dots in the mouse brain indicating the accumulation of A-beta. The system is developed in Professor Daniel Rozansky's group, and this provides a resolution around 8 micron. So the limitation of this is that it's still a 2D imaging method, so we cannot penetrate into the mouse brain. So this method has been characterized before. As you see, it can detect also the perfusion in the mouse brain and observe the vessel clearly, the major vessel, basically, the middle cerebral artery, for example. And so here we also use the APPPS1 and the Arctic A-beta mice. And we can see clearly the green dots indicating accumulation of A-beta in the transgenic mice compared to wild-type mice. And one special feature of this LCO is that the signal increased with time. Instead of like flushing away, it stayed in the mouse brain. So that's why we see it like remain very high at 2 hours after injection. So I think I'm almost towards the end. So for the ex vivo validation, we collaborate with Professor Adriana Agusi's group and Professor Hamchan's group using the mesospin and LCO, which is a combination of HFTA and QFTA to map the 3D distribution of amyloid accumulation in the Arctic beta mouse. And so the setup and the detail of this method is on the website below. And it's like so basically we can see the distribution at 3 micron resolution without sectioning the mouse, which greatly save our time and validate the finding. Okay, so in summary, I hope I convinced you that multimodal imaging provides insight on A-beta tau distribution in the brain in animal model and provide a tool for non-invasive longitudinal monitoring. And finally, I would like to thank my collaborator, especially Professor Daniel Rozansky, Professor Roganish, and Professor Roland Rick, and Professor Aguti, and Professor Nielsen, and Professor Nordberg, and the funding agency. Thank you very much.
Ruiqing, thank you very much for a very nice talk and lots of interesting techniques. Any questions? And I would like first to start with one question. What's the best resolution you can achieve with your tomography method?
So the best resolution, so far we can use 100 micron resolution, but there are like other colleagues, they use a special like a localization method can improve it even to 20 micron.
Okay, thank you. Any other questions? I have another question if there's no question. Regarding your cheese compounds, how easy it can go through a brain-blood barrier?
Yeah, so the imaging probe we use, they are small molecule below 500. So they are very easily penetrate the brain. So there will be no problem in that. So normally we check for like the suitability of passing blood-brain barrier in the beginning of choosing a tracer.
Mm-hmm. Okay.
Okay. So I saw there is one question on specificity of the stain to amyloid structure. Yeah, I think that's a great question. So, I think so the staining we use, they are actually, yeah, actually they are not specific. As you see, many of the compounds they bind to both A-beta and tau and alpha-synuclein and other amyloid like a beta sheet structure. So the aim or the major work of many group or many people is to develop specific imaging probe that detect one type of kind of fibril. So it will be depend on which probe you use.
I also have a question for you about probes against TDP43. I know that you spoke about amyloid-beta and tau, but do you have this coming? Because this is also commonly observed to deposit in brains in a large number of neurodegenerative diseases.
Yes, I actually don't study that. I only currently work on alpha-synuclein, not on.
Oh, TDP43. I was just curious whether because typically you can you probably know the problem that they can be mixed, and you're just talking about that the pathologies, they are not just pure pathologies unless you have a familial case. In the sporadic cases, you see really mixtures alpha-synuclein. Also, TDP43 can occur in a few.
Exactly. So there's a lot of comorbidity in the patient. So so what I actually so when I apply for brain tissue from brain bank, I mean, it's listed A beta, tau, and synuclein, like Lewy body status. They didn't have any comment on the other pathology, so that I cannot choose, I think.
All right. I was just curious about it. Yeah, but this is really impressive that you can push the limits of resolution of your imaging method so well. Really nice work.
Thank you.
I think for the sake of time, we will be going to our second speaker. And Dr. Ni, if you don't mind to stay for questions, there might be some more coming at the end of our seminar series. And hi, Francesco, can we test your, can you upload your PowerPoint presentation while I'm doing the introduction for you?
Yeah, thank you. I think you should have it now.
Oh, you're fine. Just keep it this way. I need a few minutes to introduce you.
Thank you very much.
It is my pleasure to introduce Dr. Francesco Ruggeri, who is an assistant professor at Wageningen University in Netherlands. Before his appointment, he completed an independent junior research fellowship at the Department of Chemistry at the University of Cambridge and Darwin College, all of these in the UK. And then he did his PhD studies in biophysics at the École Polytechnique Fédérale de Lausanne, EPFL in Switzerland. Dr. Ruggeri has led the development and the application of single molecule scanning probe microscopy coupled with different spectroscopic methods such as AFM and FTIR to characterize various biomolecular processes and biomaterials at the nanoscale. The Dr. Ruggeri's present and future research is the development and application of novel physical methods at the interface with chemistry and biology. And I don't want to take any further from your time. You can start now, and nice meeting you.
Thank you very much. Thank you for the nice introduction, Magda, and a good evening or morning to everyone. Today, I would like to just give you an overview on the research that we do here at Wageningen University on studying amyloid formation and characterizing the process one molecule at a time. And overall, I would like just to show you a journey how we moved from just trying to visualize amyloid aggregates at a very different scale that you've seen in the previous talk, because now it goes towards a single molecule scale. And from visualizing them to also be able to touch and mechanically probe them, as well as to taste them, and so to probe their chemical properties to understand how they are involved in the biomolecular processes related to disease. And this is in general important. What we know is that the biomolecules determines the rule of life, And we know that there is a very important correlation between the chemical structural properties of biomolecules and of proteins in particular, and their cellular function or malfunction. We also know that it's difficult to study these at a single molecule scale because these species have nanoscale dimensions. And in the case of neurodegeneration, this is extremely important because on one side, we have a single protein that misfold and start to misbehave in our organism, and they start to aggregate. And this leads to the formation at the beginning of intermediate aggregates that then further propagate into amyloid fibrous. What is amyloid? These are fibers with nanoscale cross-sectional dimension and micrometer scale length. Key feature of these amyloids, however, is not just the fact that they are fibrillar, but also that there is a chemical structural rearrangement going from the native protein to the misfolding state to the final amyloid 1. And in particular, we have what is called a unique cross beta sheet signature that is characteristics of amyloids. And what is important also to note is that there are different cross beta sheet signature, for instance, given by parallel or anti-parallel beta sheets. And there are several evidence in literature that show that these differences can be related in a different manner to toxicity, as well as the shape and the properties of these aggregates. So it is important to study them one molecule at a time. What we do in our laboratory, we use scanning probe microscopy and spectroscopy in combination with microfluidics, biosensors, in order indeed to study biomolecular interaction, protein self-assembly, but also organic biomaterials. And today I will show you how I will focus mostly on how we use scanning probe microscopy and spectroscopy to study protein self-assembly, and only at the end showing you a bit how we use these to study biomolecular interactions. Of course, when it comes to technical work, it's important to have a good facility in order to do this. I've been doing most of my work on protein aggregation, starting from my PhD, indeed, through my work in Cambridge with my collaborators, such as Professor Knowles, Vendruscolo, Dobson, and before Professor Dittler, but also Mezzenga. So there is a mix of people who we work with. And upon my joining in the Netherlands, together with Professor Zuilov in organic chemistry, and I work both there in physical chemistry, we have developed a facility where we can do chemical characterization from the micro to the nanoscale. I'm saying this because we are very happy to collaborate, and today I will show you an overview of which are our capabilities. So please do not hesitate to contact us if you see that what we do could be useful for you. And while I'm here, I also take the opportunity to say that we have open PhD positions, and we will be very happy to receive candidacies. So now, I say that it's important to study one molecule at a time, how protein condensation and also aggregation happen. Because, as I said, it's very important to understand how the different chemical structural properties of these aggregates could relate to the molecular processes related to the onset of diseases. But how to do that? And as I mentioned at the beginning of my talk, a possibility is think, looking at these biomolecules. So like Feynman said when first biotechnology started, it's very simple to solve any problem in biology. It's sufficient just to look at the thing, where the thing indeed are these biomolecular processes I showed you before. But also, as I already told you, they have nanoscale dimension. We are limited by optical diffraction limit of light often. And so we have need of single molecule imaging to understand the single molecule properties of heterogeneous system like protein aggregation. And one first possibility to see at objects at the nanoscale is the use of atomic force microscopy to acquire information of protein aggregates and amyloids with some nanometric resolution at the single molecule level. These techniques work similarly as it will work if there is a person reading in Braille. There is a sharp tip, the finger of the person, and there is a surface. And by feeling the interaction between these sharp tips and the surface, it's possible to reconstruct a 3D map of morphology on the object that you have on your surface, such as, for instance, in this case, we are seeing alpha-synuclein monomers. Then this 3D map is represented as a 2D color map, where the Z map is, the Z scale is represented by a color scale. And as you can see now, we have an error or a baseline noise that is in the order of 1 Armstrong. And so we can very easily, and even smaller, we can easily differentiate single alpha-synuclein monomer. And for instance, what we can use this resolution for is to monitor the self-assembly of proteins. In this case, as example, I'm showing you how in a few series of articles, we have used atomic force microscopy to monitor the aggregation of polyglutamine-based diseases proteins, such as huntingtin. In this case, we were monitoring the aggregation of huntingtin as a function of polyglutamine expansion that is genetically related to the onset of the disease, and as a function of time. So by acquiring at different time points the morphology of the sample, we can monitor how each mutation is affecting the formation of fibrillar oligomeric aggregates. Then, by looking at very high resolution maps, we can inquire the morphology of single monomers, of oligomers, of protofibrillar aggregates towards the fibrillar one. And this we have done it more than in the simple case of huntingtin, but we have done it generally in our research in the case of huntingtin, A-beta, alpha-synuclein, and other FAS and other aggregating proteins. Our interest rely in then using this high-resolution morphology information to understand amyloid formation. And another example I give you here is the characterization of the morphology of different mutants of alpha-synuclein fibrillar aggregates. So we know that alpha-synuclein is involved in the onset of Parkinson's disease, but we also know that 1% of cases of Parkinson's disease are related to genetic mutation of the alpha-synuclein protein. So here what we did, we compared the morphology and the ultrastructural properties of amyloid fibrils of the single mutation by looking what fibril at a time, studying, for instance, the periodicity of this fibril, and by using methods such as Fourier transform and hierarchical artificial intelligence, we could differentiate the polymorph that each one of these mutation creates in the fibrillar aggregates. And in the article that I don't show you here, we relate this to the different THT kinetics and microscopic steps, such as primary and secondary nucleation that can happen for each mutation. Moreover, we have used this to study the interaction of amyloids with natural products or with lipids and cholesterol, as well as we use this high-resolution approach to study oligomers in vitro, but also in human biopsies, such as brain, CSF, and blood, in a series of latest publications we had. And again, I give you here an idea of what we do, of our capabilities, because I think this is a good community to talk, also to create a collaboration and stimulate discussion. However, looking only at the morphology of these aggregates is not sufficient. And this is because, as I say, there is not only a morphological problem, but there is also a chemical structural change that occur upon misfolding and aggregation of proteins. So another possibility beyond only imaging is the capability to use a AFM and a flexible cantilever where the tip is mounted as a force mechanical sensor. So this means that similarly as your doctor will do while he's trying to squeeze your chest to understand if everything is fine or not, the tip can squeeze these aggregates to measure their mechanical properties. So this is exactly how it will look like. We have some amyloid aggregates on a surface, and then what we can do, we can apply some force with our IFM tip to squeeze a bit the fibril. And by measuring this deformation, delta, what we can do, we can retrieve, knowing the deformation that we are applying on the fibril and knowing the force that we are applying, we measure the Young modulus of these aggregates. And this means that if we do this at every pixel for which we acquire the morphology map. This allows also us to acquire maps of Young modulus, so of stiffness of amyloid aggregates, allowing so to measure how much they are stiff. And the key information that we know that is well known in literature is that amyloid aggregates have stiffness in the order of gigapascals. So they are only order of magnitude less stiff than iron. And this is related to an increasing network of intermolecular hydrogen bonding and beta sheets that is forming during the aggregation. And in particular, what we could already prove several years ago, that going from the monomeric to the oligomeric protofibrilla and amyloid fibril state, this formation of cross beta sheets and this intermolecular hydrogen bonding and contacts between the beta sheets causes a continued increase of the stiffness of these aggregates. So also this meaning that different aggregates have different not only mechanical properties, but also chemical structural properties that might relate to disease. However, as I said, doing only single molecule microscopy, so only doing nanoscale imaging, heterogeneity, measure stiffness, is not sufficient to understand which are the chemical structural changes that occur upon misfolding of a native protein, changing of structural conformation, and the final formation of this cross beta sheet signature that is still key, we know, for disease. And in order to understand what is happening to these aggregates, historically, during our path, during our research, we have moved towards a third dimension beyond seeing and touching nature, and is the concept of testing, so having chemical recognition power. And a possibility to have chemical recognition and identification of the bonds and the structure of biomolecules is the use of vibrational spectroscopy, which is very old, but also very valued. Vibrational spectroscopy and IR, for example, allows chemical identification. We can have analytical determination of concentration, as well as we can determine structure of most bioorganic materials. This is particularly true in the case of proteins, because what has been discovered in the last decades is that the COO stretching of the amide bond and this amide vibration of proteins is related, is called amide band 1, and is related to the secondary structure of the protein that we are investigating. So this means that by measuring this amide band 1 between 1700 and 1600 centimeters minus 1, it's possible to say which is the structural conformation of our protein. So this is very interesting to study protein aggregation and to understand how to a morphological change is associated a chemical change. However, the problem is that although we can distinguish in a spectrum the differences that occur between monomers and fibrils, it's very difficult to understand how these are related to the properties of single molecule species inside. For sure, what we can say is that by comparing these spectra, that monomers, they have a peak of this amide band 1 that, for instance, is related to intrinsically disordered structures or alpha helix, while when we have fibrils, we have a signature at 1625 approximately that is related to intermolecular beta sheet. So this IR can be used to differentiate monomeric versus aggregate state quantitatively. However, again, as I said, we have a very heterogeneous process, and when we use bulk methods, we cannot have information on heterogeneity and properties of single species. Now, what is our aim? Our aim is to expand this nanoscale perception just beyond seeing, and in particular, to develop methods that allows us to see at the same time our sample, to touch it, to measure its mechanical properties, but also to test it, and so having chemical recognition power at the single molecule level. And to understand, to explain better how much is important, I will go beyond just studying protein aggregates. Let's suppose to have a mental experiment, and we have two samples. One is done of protein aggregates, and one is done by DNA. We can acquire high-resolution imaging maps of these aggregates and these DNA, not only by atomic force microscopy, but in general by many sub-resolution techniques. And we can even characterize heterogeneity. But then let's suppose to mix these samples together in the test tube and put together them again on the surface. Now it becomes much more difficult to distinguish what is a protein aggregate and what is a DNA here, because we are chemically blind. As I show you, a possibility to understand which are the molecular components of our sample is to acquire chemical spectra. And for instance, we can see that we have the amide band 1 of protein that is related to the structure, as well as we have the phosphate group's absorption of DNA. However, if we will try to acquire a map of this sample on a surface by IR, we will be chemically blind. We will be, spatially blind. We cannot measure single molecule properties. The solution comes in bringing these worlds together. So bringing together the world of single molecule microscopy with the chemical recognition power of analytical methods. And so by bringing together, separating the wall, destroying the wall between these two approaches, it's possible to expand our nanoscale perception beyond seeing in order to characterize protein aggregation. And this is possible through a technology that in the last 10 years we have been developing that is called infrared nanospectroscopy. This technique now enables to use AFM, atomic force microscopy, to measure the morphology of a sample at the nanoscale. Then, as I said, by using the cantilever as a mechanical sensor, it is possible to measure the Young modulus, the stiffness of protein aggregates. But then, adding a third dimension, adding IR light, is possible also to acquire chemical maps at the nanoscale. To see how this is possible, how it's possible to have multimodal correlation of morphology, mechanics, and chemistry, I will show you another slide where I will explain you which is the principle of function of this technology. When our tip is contacted with the sample, we can send IR light. When we send IR light, if this IR light is absorbed by a sample, we will have a chemical vibration. We will have, a vibration of the bonds which are absorbing the light. This vibration can be dissipated as thermal heat, and this heat will cause a thermal expansion of our sample. And this thermal expansion can be measured by the tip of the AFM, and this response is proportional to the IR absorption. So this means that now we can have maps of chemistry together with maps of morphology. And in the same way, as I showed you before, we can have mechanical maps. Then I can stop at a specific position on my aggregate. I can record this cantilever response as a function of the wave number, and this will give me an IR spectrum, but now at the nanoscale with 10 nanometer spatial resolution, as I will show you. And in particular, by looking at amide band 1 of protein and protein aggregates, it's possible to determine their structure, as I will show you in the next slides. So this means that at the same time, we can acquire the morphology, the chemical structural properties, and the nanomechanical properties of protein aggregates. And one thing I still need to mention that even before to acquire our measurements, we also rely on advanced preparation methods based on microfluidics in order to preserve the molecular conformation on our sample on a surface. And in particular, what we use a spray that we can deposit our sample on a surface as small droplets, which dry very fast in order of milliseconds. And this super fast drying allow the molecule to not modify their conformational state upon drying. Then, once we have this technology to study at the same time the morphological changes that occur during protein aggregation and the chemical changes associated to protein misfolding and amyloid formation, we can apply these to study amyloid and protein aggregation one molecule at a time. And so what we can do finally, we can take a single amyloid fibril and characterize simultaneously the morphological, mechanical, and chemical properties. So this is the image except for a small white mistake here that you saw in the graphical abstract of my talk today. So first concern we had when we wanted to study protein aggregates, we wanted to be sure that we could push this infra and nanospectroscopy technology down to the single molecule scale and be able to determine protein secondary structure down to the single protein level. So we played with known proteins such as apoferritin, thyroglobulin, and we deposited them on our surface. Then, as I showed you, we could acquire simultaneously morphological and chemical maps that were correlated very well and gives us a spatial resolution in the order of 10-15 nanometers typically. Then what we did, we used the amide band one of these proteins and we compared them with the one of the proteins in FTIR, so in liquid environment, to measure the secondary structure. By what we do, we take this amide band one, we deconvolve it by second derivative analysis, and each peak in the second derivative corresponds to a secondary structure contribution. And what we could demonstrate is that we can determine secondary structure of protein at a single molecule level with infrared nanospectroscopy. Then what we also try to push is to have this secondary structure determination on protein aggregates, not only in vitro, but also in liquid, so to mimic physiological-like environments. And what we could demonstrate that, for instance, we could measure diphenylalanine aggregates that are the co-recognition model of A-beta 42, and that we could measure these fibrillar aggregate secondary structure in liquid. Once we had these capabilities in the ears, we have used them in order to characterize the heterogeneity and deformation of amyloids. So what we can do, we can point our tip at different protein aggregates on a surface. And what we can retrieve IR spectra going from monomers to oligomers to fibrils. And we could prove at the single aggregate scale that there is a beta sheet, cross beta sheet increase during amyloid formation, as well as that there are different pathways possible, and some of them are nothing else than protein condensation that has and liquid-liquid phase separation that are rose in this year. So we have also used similar approach to study the condensation of proteins such as FAS and to study their relationship with the onset of diseases such as amyotrophic lateral sclerosis. What we know that when a protein condensates in neurons undergo a mutation, for instance, they cause diseases such as ALS, but they cause also in the neurons the formation of fibrillar hydrogels. And what we could do with our technology, we could characterize the morphological and the chemical structural differences of droplets that are wild type, are not related to disease, versus condensates, which start to elongate, they start to have a fibrillar shape, and the cause they are related to neuronal malfunction. And what we could prove that this neuronal malfunction was related to the presence of antiparallel beta sheets, and so to a cross beta sheet signature of amyloids. Furthermore, we have used this also to understand how we could go from fast droplets and condensates to fibrillar materials that similarly as the ones that are forming in the brain of patients, and which was the droplet to fibril structural change that was occurring. And by looking at by using polarized light and nanopolarimetry, we could demonstrate how we were going from a dispersed state to droplet formation and final fibrillar formation of FAS. And most importantly, I will go a bit quicker now because I think I have lost really a couple of minutes. We could use the same approach to study the presence of protein aggregates in living organisms, such as, for instance, E. coli, and to prove the distribution of Carli amyloids on E. coli that can produce them more or less, and measure by looking at this cross-beta sheet signature, how much protein aggregates there were in different strains of bacteria. And of course, we have the interest to move this towards more relevant in vivo systems in the case of neurodegeneration. Finally, in the last minute, I will give you lax example of what can be powerful to use these single molecule approaches in amyloid research. Ultimately, we are interested in developing pharmacological approaches such as small molecules or antibodies that could interact with our amyloid aggregates to modify the pathways of aggregation and the onset of the disease. My colleagues in Cambridge and the group of Professor Vendruscolo, and now there is a big company had between the others by Johnny Apchke, the person who has collaborated with me on this study. They have developed small molecules which can delay the aggregation of amyloids in vitro, but they can also delay the aggregation in vivo of A beta, and so stopping the signals of neurodegeneration in vivo systems. And what was our interest here was to study how this small molecule can interact with single amyloid aggregates. So what we could do, we could measure the IR signature of these oligomers and fibres of A beta when they are alone and in presence of our drug. And then what we could see that when we are in presence of our drug, there are new bands that are appearing that are related to the interaction of the drug with our protein aggregates. And what we could do, we could apply multivariate data analysis together with artificial intelligence to prove that our small molecule is interacting with our amyloid aggregates through a single hydrogen bonding. And most importantly, that if we will remove this single hydrogen bonding, we could basically stop the power of our drug in stopping the onset of the phenotype of Alzheimer's disease in animal models. So this means that we could identify the key molecular interaction between this drug approach to Alzheimer's disease and our aggregates. And with this, I hope I just illustrate you how we develop single molecule approaches to study the heterogeneity of protein self-assembly, not at the morphological level, but correlating morphological and chemical properties to provide information that can be useful to combat neurodegeneration, for instance, developing a new molecule to target the disease. And with this, I would like to thank you all for your attention.
Thank you very much. It's a really exciting talk, and it's really a nice mesh of different techniques, all giving very useful information for amyloid formation. I have a question which I've been thinking to you. You probably have already the answer. Why do you couple IFM with FTIR? Why don't you use Raman?
Yeah, this is, first of all, any technique can be useful. It exists also the equivalent of nano-IR, that is deep-enhanced Raman. The problem with deep-enhanced Raman is that there are a few key points. That is a scattering-based techniques, and for reasons that are still not clear, the amide band 1 of protein is not always present in the nanoscale spectra. Sometimes appears, sometimes not. And this is clearly a problem when we want to have a structural characterization of cross-beta sheet signature. Another problem is that the spectral resolution itself for Raman is, we have like spectral resolution of below 1 centimeters minus 1, where Raman goes towards several centimeters minus 1 for band deconvolution structural analysis is a bit less sensitive. And ultimately, Raman relies on probes that cannot be always produced successfully, while we can use a very commercial probe, very accessible, that they don't degrade. So this means that we can have higher throughput. So this is what has made us having the choice to use IR. But of course, TERS can be very highly spatial resolved and can provide useful information as well.
Yeah, thank you. That's a good point, which I didn't think, because when you say FTIR, it's just you start like making big eyes, water can interfere with everything, but you have been drying your samples, so the water is not the problem, or maybe you do hydrogen deuterium exchange.
We can measure in water. We have demonstrated it. We can measure secondary structural aggregates in H2O. But of course, D2O is also a strategy when working with IR.
Yeah. Thank you.
Thank
I
you.
saw that Rams posted a question. If a single H bonding is kin fiber bonding, how selective is the small molecule inhibitor under in vivo? He posted it in the chat.
Yeah, I can see. Yeah, this is a key question. What I can say that indeed in Cambridge, this study on this bexarotene molecule was only really the first paper to then have a big company that is now called Varen Therapeutics, where they are studying a lot of molecules and targets and specificity towards the aggregation mechanism. So what we wanted to show here in this study was more a proof of concept that we can study single aggregate molecular interaction. And this is very important because when people try to do docking, often use structural information that is averaged, while there is need to understand how a molecule can interact differently with different aggregates. And this is a direction where I think slowly we should move towards.
Yeah, thank you very much.
Thank you.
I have another question for you. When you measure the Young's modulus with AFM, the way I see it, and again, I don't have any experience in AFM, I'm just curious. You show the picture where you measure a Young's modulus, even in the fibrils which are overlapping on the top of each other. I would think that this will be a little bit challenging to measure it on a bundle of fibrils because you don't know how many fibrils there are. You just can only measure it on a single fibril. Can you explain a little bit about it?
Sure. We first of all, looking at the maps, we have power of heterogeneity, so we can measure the Young modulus of single fibril. We don't need to measure on a bundle. We can exclude. This is the power of mapping at the nanoscale.
Yeah.
So we don't need to measure on the bundle, and we just get the information from the single fibers and we compare. So we can do single aggregates, Young modulus measurement.
Yeah.
Then still, Young modulus is an intrinsic properties that doesn't depend on the quantity of material that you have there. So if you have fibers that are identical and they are bunching together, the stiffness of 3 of them will be the same as the stiffness of 1, just because it's not an extensive property, but it's an intrinsic property of the system. Still, we don't need to rely on that. We can perform the measurement on the single fibrils.
Yeah, so I'm guessing for when you have multiple ones, you see if it's divisible by the single one, you can extrapolate to that, something like that, right?
Yeah.
Yeah. Oh, we have a question coming, and actually we have 2 questions on the Q&A.
Please.
One is from Olga Gursky. She says, nice talk. Can you have methods extract information on fibril fragility, which is important for amyloid proliferation? Very nice question.
Well, Young modulus can give information on the stiffness on the fibrils, but I think doesn't give information on fragmentation mechanism that are, I think, maybe the one where the question was referring to. What is cool that we can do having single molecule methods is that we can measure the amount of fragmentation in a system in vitro. In principle, we have started to try to do these measurements also in vivo, and now we have a series of work in collaboration with Professor Krennerman, where we study the size of fibrillar and oligomeric aggregates in human biopsies. And we can start to look at differences. Of course, in vivo is much more challenging than in vitro. So quantifying fragmentation, I think, is more feasible in vitro. But it's doable because you can look at your microscopic steps, for instance, by THT chemical kinetics, and you can see where you observe microscopic steps of fragmentation, and then you can use imaging to compare the states with or without fragmentation.
Yeah, thank you. The other question is for Camelo. My curiosity in FM measurements, how much does the substrate affect the interaction between protein and surface?
Yeah, having a surface can be always good or bad, depending on the point of view. Sometimes amyloid formation occurs on surfaces, such as lipids. Often, when we want to prove the properties of the aggregates, we prove them on independent substrates. So this means we take the same sample, we deposit on different surfaces, and we measure the same property. And we are happy when this property doesn't change with the surface of deposition, because for us, it's a good indication that it's an intrinsic property of the molecule. But of course, this is not it will be ideal always to measure something out of a surface. Cryo-EM is great for that. The only problem with cryo-EM that you need to average multiple aggregates often to get structural determination.
Yeah, thank you very much.
Thank you.
I want to see if any of the panelists have questions to the speakers. Sam, Rams, and I'm asking the audience if you have any questions, you can raise your hand so we can allow you into the panel to ask them personally.
Great talks. I have no questions.
Oh, thank you.
Yeah,
I think there is only one thing I can add. There is when we present a method to study something, there is not the perfect method, right? And everything gives you some information. It has drawbacks. And I think the power is in using different ones, but bringing new information is key, using complementary approaches.
I really agree with you, and it's really nice demonstration what you're doing. I wonder, I know that it will get a bit too complex, but if you can add fluorescence or single molecule on the top of it, but this may become kind of difficult because you need to deal yet with another station to add on.
Yeah, there are attempts to do this. At least for FM, this is done. There are FMs combined with a single molecule fluorescence of commercial systems.
Yeah,
so
yeah, no, but the three of them, kind of, if you can do that.
Yeah, is a direction where we are slowly thinking to go, but it's still in the development. I think 11 in Belgium, they have a system. They have purchased the same technology to try to study as well protein aggregation, and they have a system where they try to do also fluorescence. But I still don't believe at the single molecule level fluorescence is more having some sort of indication where are the aggregates of your target in a more complex system. And we are having the same direction, the same tools, so the same capabilities.
And I just, before I let you go, can you talk a little bit, because I this is very likely changing about the tip of the cantilever. How is the dimensions and what is the current stage of the dimension? Because this is largely what determines the resolution. If there's any dramatic improvements, let's say in the past, during past few years.
I think this is the major advance of IR compared to Raman at the nanoscale. We can use virtually any probe, depending on which is the configuration of the instrument, if we have light from the top or from the bottom. So this means that the resolution is technically the same as a FM.
Okay.
And in particular, I didn't advertise too much. I maybe forgot. In particular, the FMIR instrument that we have now is what is considered the most sensitive actually existing. And we start to be able to do chemical analysis down to a single, like for instance in HOPG, single atomic steps that we can retrieve. We have very low level of noise in the order of picometers. It's not as sensitive as TERS. For sensitivity, TERS is superior. But what it comes for versatility and having good spectra, I think now we have an advantage there. It's always a complementarity.
No, I understand it. Yeah, thank you. That was really both very insightful talks. Thank you so much.
Thank you for hosting.
Yeah, Francesco coming, and thank you, Ni, for coming in for the nice talk. And I don't see any more questions, so we are ready to finish. Yeah, nice seeing you.